Hello everyone, my name is Ashfaq Ahmed and I work in the field of structural biology and bioinformatics. I welcome you all to Bioinformatics Insights. This is part 2, or I would say the second part of the QSAR modeling analysis. I expect that you have watched part 1 and have generated your datasets for this part. If you haven't watched the part 1 yet, I would suggest to first watch part 1 and generate your dataset. Otherwise, I'm afraid you may not understand this part. Okay, so let's come to the business point. What we will study or what we will do in this tutorial, this part was covered in section 1 or video 1. And in this video, we will cover the remaining parts. We will use the data prepared in previous part. We will generate different models. Then we will test those models. Then we will optimize those models. And then we will use it for testing and training. And once we are satisfied, then we will use it for predictions. So let's start. Uh, okay. So once again, you need to type this here. I will drop this link in the description section. Then you will see this thing. Then you need to click here and select the CAD project. Then scroll down and come to the QSAR model generation and testing part 15. Why I call it part 15? Because it belongs to the drug development and drug designing. So technically I add this QSAR in the drug designing playlist. So it is part 15 here. That's why. Otherwise, in terms of QSAR, this is the second and final part. Okay, click here. Now connect here, but I would suggest first come here, then change runtime type and click here T4 GPU. Connect with GPU because we will perform some training and testing. So on CPU it may take a bit longer. And most of you will be free user because if you see here, you may not see this pro option. It means you are a free user. So according to my understanding, you can use the GPU for 4 to 8 hours. Depends on the crowd on the GPU center on the GPU server. So it's okay. Why not use it for this notebook? Okay, connect. Okay, this is once again just uh, a quick details or the quick introduction. But if you have not prepared the dataset, once again, you have to watch this video. Once this video is uploaded, I will quickly mention the link here. So you will see it here, no problem. Now click here to install the necessary libraries. We will learn side by side. And okay, libraries are installed. Now we need to import those libraries. Sorry, not libraries, programs are installed or packages. So libraries, from there we import the libraries. Okay, now here we will load the data which we prepared in part 1 earlier on. So it's present here and it is merged descriptors. Merge descriptors. It was prepared today on this time. So what I will do, I will just click here and click here to load the data. And it's here in the download section. So load it. And we have the data now here. So we can check it whether we have loaded the correct data or not. It's necessary. Okay. Click these three dots, copy path, and paste. Read the data. Now we want just if you want to see some heads of the data, so why not? So the data contains these columns, smileys, QED, molecular volume, surface area, blah, blah, blah. And all of these things are calculated in the previous video. Now here we will define feature in the data because our data contains too many columns. If we count it, so it's almost more than 10, 1, 2, 3, 4, and 5 here and 4, 5 here, almost 15, 16 columns. So we need to tell the program that which column to treat as a feature and what to not. Okay. We will tell the program that do not calculate smileys, do not take smileys column and activity column as a feature and takes all the remaining column as a feature. Okay, so here we actually define these variable. If you are interested to see the code, click here. So we ask here like feature is equal to data dot drop and drop these two columns. Do not include them in the feature. Okay, click here if there is any missing data, but it is optional. We know we don't have any missing data. We have manually checked it. So still, okay. This will normalize the data if there are so abruptly or very high values. So the data will be normalized. So you need to click here or the data will be scaled. You can say like this. Now we need to tell the programs that in next step, we will define some models, but you have to treat our data in this fashion. It means that you have to treat 80% of the data for training and 20% of the data for testing. It's a fashion in machine learning. I also talked before about this. So we are going with 80-20 ratio. Some people are going with 70-30 ratio, 60-40 ratio. It depends on your data. If you have a huge data set, if you have a very huge data set, then you can you can tweak these numbers. But if your data is not a huge one, then I would suggest 80-20 is a better option. So click here. Now our data will be splitted into 80-20. It means in every 100 rows, 80 will be used for training and 20 will be used for testing. Now we have almost some 2000 plus compounds in the data. So then you can calculate how many will be used for testing and how many will be for training. Initially, we will build simple QSAR model using linear regression and random forest regressor. So you click here. If you want to see, you can see the code and you click here. So this one is a linear regressor and then this one is a random forest regressor or random forest model. Okay, we build the model. Now we want to evaluate the model because we have already asked like split this data in this format, use all the other columns as a feature except those two. And now we are going to evaluate these two models on the data. So we clicked here and that was so quick. It says that the model gave an error of zero point 0.8 and the R square score was 0.1. Actually, I have also talked before on this in a sufficient detail that what the MSE and what R2 means. The MSE or mean squared error is actually the model error, and the R2 is actually the prediction error. Okay, so it seems fine, but um, like humans, we always search for the better. So let's see how random forests behave. Random forests behave better as compared to linear regressor. You can see the MSE is zero point 0.4 and the R2 is zero point 0.1. It means the chances of error made by this random forest are smaller compared to this. Now we want to save the results of this in a CSV file, and if you click here, the file name will be model performance matrices. Okay, so just click here. Now you can also see this print option and you must have a new file. If let's say download it and see how it looks like. So it will look like this. These are the two models we tested: linear regressor, random forest, and mean square this and R2 and R2 square this for these two models. That's it. Okay. Now we can also save the results here. The file name will be QSAR results. So refresh. Let me show you. Oh, come on. No. Download what these results mean. Okay. Now just a quick look. These are the smileys we used. These are the actual activity which was when in our file, and these are the predicted activities. Like 16.4 was the actual activity, but linear regressor predicted 16.5, a bit on the higher side here 16.8, but linear regressor predicted 16.6, a bit on the lower side. Here you can, this column is for predicted activity of random forest. Okay, so these were the results generated by these two models. Now, if you are interested to see them in a kind of visual plot, so this is the linear regressor regression results. Predicted activities I showed you in the CSV file and the actual activity, and that CSV file you can use it for your own if you know some better graphing or visualization, so you can plot it there also. Okay, and this is called the residual for linear residual plots for linear regressions. It's almost the same thing. It explains the behavior of the model. But I will not go in a more detail because there are much more to discuss, and I don't want this video to be on a longer side. So if you want to understand, you can ask from Google and you can read some paper. But it's actually a kind of matrix or a measurement for the model. Okay. Now, feature importance for linear regressor. Remember, according to with these errors, linear regressor made with this error here, it says that among those features, these were all the features we calculated. The most important feature is molecular weight and followed by the Zagreb index, followed by the kappa 2. And according to the model, linear regressor, the other features are not pretty much important, and even some are on the negative side. Okay. Now, what random forest says, because it behaves a bit better, we cite here that the random forest has generated a smaller MSEN R2 score. So it means random forest is better among these two. And according to the random forest, molecular weight is not important. You see, it's here. So now which model you will believe? Of course, scientifically, I should go with the result of random forest in comparison of these two because the error chances are almost 50% smaller. The linear regressor was 0.8 and random forest is 0.4. So these results are better. So according to the random forest, the Zagreb index, then the Bob index, and then the log P, and then QD are better, and even the QD and log P are almost at the similar level, and you can you can see this. Okay, now this was the simple model generation and simple model testing. If you believe that you don't need to optimize the model, then you should directly jump here with use save the best model and then jump here and test for unknown compound. But I'm afraid most of the time these simple models are not better. Uh, and I hope the same in your case. So you should learn the optimization strategy. Suppose these simple models are not better or we did not like, so we are in search of some better model and some better results. So let's import the necessary things. Now here what we do, here we define three more models. Now, here we define linear regressor, ridge regression, linear regression, lasso regression, random forest, and gradient boosting. And okay, let's define. And we use some hyperparameter grids. In simple terms, if you are a clever student of biology, so it means we have used some different parameters for all these models, and let's see, click it. So now we will evaluate the performance of these 4 or 5 models, and let's evaluate. So that's why I was asking you to connect with the T4 GPU, because if I was on CPU, then if it takes almost 2 minutes, in that case, it would take 5 or 6 minutes, or could be 7 minutes, but it also depends on the data. Okay, so let's see the result. Linear regressor, almost a similar result. Okay, and it says that for best parameter for okay, for ridge regression, that is zero point 0.1 and alpha zero point 0.1, because in the parameter we check different alphas. Now, the rig regression almost similar to this error, and R2 score is also the same, seems the same. Now, lasso regression results are, I think, almost the same. You can see, but the MSE score is on a better side as compared to uh, the others. Okay. Now we are waiting for other two models to be tested, and then we will save the result just like before. So let's see. I'm going to pause this session. Welcome back. It took almost 4.5 minutes. Okay. So the random forest is zero point 0.4 and zero point 0.5, and gradient boosting is another model. It took zero point 0.4 and zero point 0.5. Okay, doesn't matter if you don't understand. You can save these results, and now if you want to save the output just like before, you will see the predicted and the predicted PIC 50 by all these different models. Okay, why I said that doesn't matter if you do not understand them completely, because now we will check, or we will select the best model, but I have dropped a script below, so the computer will do this job for us. So therefore I said doesn't matter. Okay, and if you want to save the output, you can save it. If you want to say the plot actual versus predicted for the best model. So here among these five, the best model is was selected by this program was gradient boosting, and these are the results, and this is the residual plot for the best model. And okay, now we will save the best model now. So you can, so the first, once again, the same thing will be repeated again, because we will ask the computer to rerun and reevaluate, and then choose the best model. Okay, so I have to pause it again. Okay, welcome back. So the evaluation was performed, and now we will ask him to save the best model, and you will see this, that the model performance matrix is saved to model performance matrices dot best dot COE. Okay, QSAR results saved to this. Uh, okay, save best model. Here you can see all those files. Okay, the best model was saved with the name best underscore model dot pkl. You can see here. Okay. And if you want to do some plotting again, so these are the those plotting, and now see the features information. Okay. Now compare these two things. First, linear regressor says molecular weight is solely important because if you can see, they are way below than molecular weight. Then regressor random forest says no, molecular weight is not that much important. It's way down here. There are many more which are important and they are here. And it says like Zagreb index on top and bulb index on second and log P on third. But then we perform some optimization and some more included some more models. What we achieved that the Zagreb index and kappa 2 and then number of rings are important as compared to the bulb index. Bulb index is important, but it's here. Why we believe gradient boosting? Because in the evaluation among these five, it behaved well. So here I want to share, for the sake of time saving, I have just included these five models. If you are a bit smarter, so or I expect you to be smarter enough, so you can include more models. Now, if you don't know what are the names of other models, so why not ask Google? Like I have tested these models, I want to test some more. So just include in the script. So in this case, you will test many more models. Okay. Now, and if you want to stay happy with these five, so it's up to you. Okay. Now, here is the business end. Before going to this end, I just want to remind you what we have done, or to refresh your memory. We pick the data set, we calculated the descriptors. Remember, you can also calculate more descriptors. And if you calculate more descriptors, although 15, 16 we have calculated, but you can increase them. And if you increase them, ultimately, statistically, your results will be increased. Your results will be improved. Now, then in this part, we first define some simple models, then we check their evaluation and analysis, then we went for five models with some parameter settings, and then we evaluated those five models, and we found the best model among them, and we load that model here. Now, why we have done all these things? Because we want to perform a kind of virtual screening before docking. The term virtual screening, normally our student, they take it as like you take a bunch of compounds and they then dock it and it means virtual screening. That is correct. You can go for virtual screening on pharmacophore based and structural based. That are another avenues. But here we will perform a kind of virtual screening for the unknown compound. Now, here what we will do, we will load the best model. It's here. So just load it. Okay. Now import the libraries. Now, wait, what happened? Okay. It says that RDKit is not installed or whatever. So we can do it. Pipe install RDKit pipe. Okay. So we are okay. Now import library is done. Now here they are asking for a new or unknown compounds. Technically, I do not have unknown compounds, but what we can do to test it for you, I will use the same thing. This is our merge descriptor list and we use it initially. Okay. What I will do, I will remove this activity column. Let's say delete. Now you can see our data has each and everything smileys and the features and no activities. Okay, close it. Remember, if your if you have your data, so it means a data of unknown compounds. I mean, if you have your data, so it means you have smileys or you have Kimball or Zinc or whatever IDs. Now you have nothing else. So what you have to do? It is must that you have to first calculate all the features calculated before during training. So what you should do, take a list of unknown compounds and then rerun the QSAR1, calculate the descriptors and bring it here and directly import the best model. You connect here and it doesn't mean if you don't want to run these things because you have already run it and you have the best model in your computer, so just click here, load the best model pkl here and start from here. Okay, so if it gives you error, so starts from the installation of rdkit 5p, then do this. Now here they will ask you to load the new compounds. So we just suppose or we suppose that this is our unknown compound because I have removed the activity column here. So these are our smileys and these are those features and no activity. These are the some random unknown compounds. Let's say I will name it, let's say, not to confuse, I name it unknown. Now assume these are our unknown compounds and yet they are unknown because uh, we removed the activity column. Now let's see, click and upload it here, unknown. Now once it is uploaded, then we will click here and we will copy path, show code. Oh, sorry, not for this, for here, show code and drop it here. Extract feature column, assuming all column except smileys and activity are features. Okay, although we don't have activity, so that says, okay, scale. Now perform predictions with the best model, add predictions to the data frame, save result to the best model, and we will change the name to uh, unknown result for unknown. Okay, save. Let's see, a new file will come here. Result for unknown should be here. Why I can't see? Yes, here. Predicted activities of unknown. Okay, predicted activities here. The name is here. Okay, so download. Now see, these were our unknown compounds. So every for every compound, they have predicted uh, some activity, and the error was on the minimal side, so almost originally I remember it was 16.4, now it's 16.5. This was 16.7, this is 16.8. At least, at least, if you select the better model, then you will receive a kind of near to the accuracy. Your result will be a kind of accurate. Now, what is the use of QSAR screening? If this is your unknown library, almost 2000, it's hard to calculate or to dock each and every one. So why not use this feature? Click here for, for click here, sort them with the okay, sort Z to A. Now you see 19 is the top value, 19.6. So why not select only these 19 for docking? Isn't a good idea? Okay. Now how you will say that these 19s are on the active side? In our last video, not of QSAR, in the exploratory data analysis, if you remember, I focused on a one figure and I told you that it is the important figure. The figure was very simple. On one side there were active, PIC of active, and on the other side the PIC of inactive. Okay. So if you compare these results with those figures, so you will know whether 19 comes in the active range or 19 comes in the inactive range. So now you have options where to go or which one to select. Okay. So, okay. Now it was the testing section. The final and important section we will discuss, we will take 4 to 5 or 6 more minutes. So stay with me. These are important. Research directions and applications. I I discussed this thing like virtual screening. You can use it for virtual screening, but remember, virtual screening, you can perform virtual screening with pharmacophore, with structural based. Most of you are aware of it. You can also perform virtual screening with QSAR. Today we have done it. You can also perform virtual screening with artificial neural network. We have done it in video number 11 in the CAT course. This is 15. Okay, go there, and artificial network is something more powerful than machine learning. Okay, so I taught you three different things in this course to perform virtual screening. Now, assuming that you apply all these three things in your data, and then you select some potential compounds for docking or for lab setting to be tested in lab. So you just believe what level of their study will be. So normally our students, they just do some docking and then they say this is inhibition. So don't mind, but I call it a joke. Um, and normally then we are saying that our papers are not going in good journals. So because we are just taking some sand and some bricks and then we mix them and then we say, look, the sand is stick to the bricks. So of course, the sand or the mud will stick to the brick, but how it is stick, we don't care. So first, you need to analyze the brick, how to keep, and then we will use the mud, where to to apply that mud. So this was the simple example I could give to you. Okay. The second thing is exploratory data analysis. I will not repeat the same information here, but I can guide you. If you remember, we use the data set from the Kimball and we perform exploratory data analysis. But after performing the QSAR part 1, your unknown compounds will have all these features. Let me open it again. Your unknown compounds, because we call it unknown, so your unknown compound will have all these features. So why not go there and repeat the EDA or the exploratory data analysis for all these features? What we have done there in EDA, we just use Lipinski rule of 5. It means we had 4 columns: log P, molecular weight, number of hydrogen donor, and number of hydrogen acceptor. Now we have for the PIC 50, we have the predicted column here. So the thing is, I did not save it, I guess. Okay, I opened the unknown, so that was in the predicted. So why not to perform the exploratory data analysis? So this is another result for your manuscript or thesis or paper. I'm just giving you, or I'm just exploring different avenues in your mind. Okay. The last and the final questions, that compound library of unknown compounds generation. Okay, some spelling mistake or construction mistake. I will fix it. How you will generate a compound library of unknown compounds for your research? That is important. Frankly speaking, today I have wasted almost 2 hours. For what? Because I tried different things to scan a database, Zinc or PubChem. I tried different things to scan a database and collect the compound based on the Zagreb index, kappa 2, and all these features. Still, I'm looking for it. If I get success, then I will share it to you. If I can bring that thing, so the compound library generation will be easy for you. You will not randomly pick compounds. For example, if in your analysis the Zagreb index is on the top, then why not use the Zagreb index information? Let's say check this file, and if the Zagreb index is important, so the Zagreb index starts from 254 and the lower value is from 168. So why not use the range that the Zagreb index from 168 to 254 something, and go into the PubChem and then download all those compounds which satisfy the Zagreb index in this range. Wouldn't it be exciting? That will be your unknown compounds, a library of unknown compounds. But after not wasting, actually, I'm doing this all the day. So I'm still looking for it. And if I found it, I will share. So then you will be able to generate your unknown library of unknown compounds based on features suggested by the machine learning. So you will work with more targeted stuff. But so far, if I am unable, so then how to generate the question still remains the same. Because there are millions of compounds in different databases, it is impossible to treat all of them. In 2000 compounds, you saw that we were waiting for 10 minutes or 5 minutes. Now, suppose if I was giving you this tutorial with 1 million compounds, how would that be? So it's almost computationally, it's a kind of foolish job. So what should we do now? So I have another idea I want to share with you. There is another video like search for similar compound, and I know that video got very little hits or views, but I'm not sharing it for the sake of views. I'm sharing its importance. Why not? If you have, let's say you have some receptor for cancer, and the receptor is working in lung cancer, let's say, then lung cancer or another cancer. Now, go and search for the FDA approved drugs against that receptor. You will find these information. So let's say you found gefitinib or something else, let's say three or four. Now, why not use that search for similar compound and repeat it for all those four, five, six compounds and reduce the similarity threshold, let's say 50% or 60%. It means that you will be able to receive a diverse similar or diverse compounds. Because if you keep the similarity index up to 98 or so, almost the similar compounds. Okay, now, so let's say you have four FDA drugs for cancer against this your target. Repeat that notebook with 50 or 60% similarity and download, let's say 1000 or even more if you are interested. So your total library will becomes 4000 compound. Now bring it here and calculate the first feature. The in a previous video where uh, the Zagreb was there. But if you have the Zagreb or if anyone, if you have, so just calculate the features for that 4000 or 5000 compound. Now you have your data, check your data which you use in training and go specifically for that feature. In my case, it was Zagreb. So go to Zagreb and the 254 and then 168. So in those 4000 compound, if I'm you, what I will do, based on Zagreb, if compound Zagreb is below minus 68, below 168 or above 254, then I will discard it. So that way I may have a compound library of, I would say, uh, 500, 600, 700, 1000, whatever. Now, what I should do, I test it here or test it on the artificial neural network. So, uh, the way I told you was the selection of unknown compound is, I feel it is more scientific to select a diverse group just randomly. So why not select a compound based on a feature? So these were all the applications of this videos or the NN videos, artificial neural network, and I have already covered much more things. So finally, a closing remarks. Uh, I think formally I should announce that this CAT course is finished now. Or maybe I who knows, maybe I add more videos to this CAT. But so far, I think I have uh, gave you how to filter, how to generate, how to use machine learning, how to use artificial neural network, how to do QSAR, and how to do docking with offline, with online setups. So I think majority of the angles I have covered here. And if you are liking my work, so I have just one request to subscribe and share this channel. Um, the rest is up to you. Thank you so much. Stay happy and bye bye.